I'm Dennis Piehl. I'm a scientific software developer at the RCSB Protein Data Bank based at Rutgers University. Following from Jonica's presentation on the KBase workflow and how it makes use of PDB data, I'm now going to transition our focus specifically into the PDB archive and the structural data that's available there, as well as the tools that are available for accessing that data through the RCSB PDB. So I'll first begin by giving an introduction to what the PDB archive is, as well as who the RCSB PDB are. I'll then give an overview of our search tools, as well as some of the available download services that users may use. And I'll also give a discussion on the types of formats that structured data can come in. We'll then move into a live action tutorial in which I'll demonstrate some basic procedures on how to access data from rcsb.org. And this will focus using the case study of the Arabidopsis thaliana resistosome. So some things I'll cover are the basic searching and browsing of our data, looking at structure summary pages and exploring the data that's available there, how to download the data in various ways, as well as the programmatic manners of performing the searching and downloading tasks. And then at the end, I'll conclude with some additional resources and documentation that you may look at for reference on your own. So first, to provide a brief background, the Protein Data Bank, or PDB, was established in 1971 as the first open access digital data resource in biology, containing just 7 X-ray crystallographic structures of proteins. Since then, the PDB archive has grown to house more than 190,000 experimentally determined 3D structures of biological macromolecules, including both proteins and nucleic acids. These structures are determined experimentally by tens of thousands of researchers around the world using techniques such as X-ray crystallography, electron microscopy, nuclear magnetic resonance spectroscopy. And then they are submitted for deposition to the PDB archive, in which they undergo a thorough validation and biocuration process before they're officially integrated and made released to the public. This PDB core archive is jointly managed by the Worldwide Protein Data Bank, or the WWPDB, which is comprised of the RCSB PDB in the US, where I'm based, PDB-E in Europe, PDB-J in Japan, BMRB in the US and Japan, and EMDB in Europe. So while the RCSB PDB, PDB-E, and PDB-J provide access to the structural contents of the core archive through their respective websites, the BMRB and EMDB provide access to the NMR and electron microscopy data through their sites. We are committed to making sure that data adheres to FAIR principles, that is, that it remains findable, accessible, interoperable, and reusable, as well as ensuring that data is made freely available to the public with no limitations on usage. The RCSB PDB web portal at rcsb.org provides not only access to the contents of the PDB core archive, but also a rich collection of tools and features that you can use to search, browse, analyze, visualize, and download the contents of the PDB archive. Additionally, on a weekly basis, we furnish the collection of structures in the PDB with annotations from approximately 50 external data resources, such as UniProt, SCOP-E, CATH, and CIFS. And these provide additional biological and evolutionary context to the structural content. So we are always working to continuously add and develop new tools and features at rcsb.org to enhance the user experience and to give them more ways to explore our data. For example, we've recently introduced a 1D, 3D protein feature view, which allows you to map residue-specific annotations from the 1D sequence directly onto the 3D structure, all through an interactive visualization tool. Additionally, this has been recently extended to groups alignment feature, which will allow you to align multiple proteins on top of each other. And both of these features will be demonstrated in subsequent talks by my colleagues, Brenda and Shuchi. So one major new development we've recently introduced that Stephen described earlier is the integration of approximately 1 million computed structure models from AlphaFold 2 and Rosetta Fold into rcsb.org, which we are now delivering alongside the collection of approximately 200,000 experimentally determined PDB structures. This integration tremendously expands the coverage of protein structure space encompassed at rcsb.org, filling in gaps in available structures for proteomes that were previously underrepresented by experimental data alone. And Brenda will discuss this further on. Last, alongside the data, features, and tools provided at rcsb.org, we also offer an extensive collection of educational and training resources to both aspiring and experienced structural biologists alike. And this is through the PDB 101 RCSB web portal. So when you navigate to rcsb.org, you have a couple different options for searching. The first is this top bar basic search. And this serves as sort of a Google-like type searching functionality. So you can enter in any set of terms, you can enter in a specific entry ID, or even an amino acid sequence. And when you hit the search button, you'll get results that are ordered in order of relevance. You can also fine-tune your search using the advanced search tool or advanced search query builder, where you can specify specific attributes or different qualities about the structure that you're searching for. And I'll give a brief overview of each of these in a second. The last thing I want to point out is you can also, if you're interested in programmatic usage, you can also click on this JSON button here once you perform a search, and this will generate the JSON query that you can submit to our search API, and you can integrate that with whatever programming tool you prefer. So to give a brief overview of each of the advanced search services that we offer, the first is the full text search. So this is, again, that full text Google-like search that I described, where it looks for structures that contain the terms that you search for and ranks them in order of relevance based on how frequent those terms appear. For a more fine-tuned control of your search, though, you can use these other search services. The first is structure attribute search, where you can search for structures based on a specific property about the structure. So you can look at all structures for a specific experimental method, like electron microscopy or X-ray crystallography. You can search for structures based on the author or the size of the protein. And we have a large variety of different attributes you can choose from. You can also search for structures that contain specific chemicals. So if you're looking for a structure that contains a particular ligand or cofactor, you can search for that using the chemical attribute search, or you can specify the name or the weight of the chemical. Sequence similarity search, as the name implies, you can look for structures that have sequences that are of a certain identity threshold to an input sequence. So you provide an input sequence and specify what cutoff you want for how close those sequence will be, and it'll return all the structures that have a similar sequence. Most of you have probably used a BLAST search at some point, but we use the MMSeq2 algorithm. You can also perform sequence motif searches, which is looking for a particular pattern of amino acids within a sequence. So let's say you're looking for all sequences that contain a serine, threonine, wildcard, glycine combination. Well, you can use this regular expression string in your search to find all sequences that contain that. The structure similarity search, as the name implies, you can look for structures that have a certain global structure similarity to the structure that you're querying. So you'd provide an input entry ID and chain ID to look for proteins that have protein chains that are similar global structure. You can also do this at a local level using the structure motif search. So if you're looking for a certain binding site, like the residues that comprise a particular binding site, you can search for that using the structure motif search. The chemical similarity search lets you look for structures that contain chemicals that are of similar structure to the input smiles or inchy strings, as well as you can even sketch your structure directly using the draw tool. You can choose your return type as well. So by default, structures are returned as entries. So this is the entire entry composed of one or more protein chains or nucleic acid chains. I shouldn't be strictly talking about proteins. You can also choose to return specifically polymer entities. So this would be either a unique protein chain or a unique nucleic acid sequence. You can also return your structures as assemblies, or you can get the non-polymer entities, and you can also get the molecular definitions. So molecular definitions refers to basically these chemical structures, the chemical definitions. And you can also consolidate your searches by grouping results. You can group them by various criteria. For example, if you want to, you can group them by sequence or of a certain sequence similarity. You can group them by deposit ID and as well as uniprot ID. So this allows you to more cleanly sift through your search results if you have a lot of them. So I mentioned earlier that if you were to click on that JSON button, you are brought to a Search API query builder. And basically this will just present you with the JSON object that you would use to search our Search API service. And so you can customly build your query within our UI and then click on the JSON option in order to see how that query would look in your API call. And in fact, this is how the back end of KBase is working when it interacts with our website as well. It's using our APIs. So in addition to multiple search services, we also offer a couple of different ways of downloading data. So first of all, while you're browsing rcsb.org, you can download data directly from a structure summary page or an entry page. You can download all the data or all the structures from a search result page. So if you get a list of 10 different structures, you could download them right from that page. And you can also use our bulk download tool, which lets you enter in multiple IDs to download. Alternatively, you can also navigate our PDB archive directly or access it programmatically. So over HTTP, you can access our PDB archive at s3.rcsb.org, which is an Amazon S3 bucket of our PDB archive. It's also available at files.rcsb.org. And you can use any command line or scripting tools to access this, so curl, wget, or Python. And since the theme of this workshop is structured data, I wanted to emphasize that the structured data is located under this directory here. I'll give a quick demo of how to get there too, but this is where you can find a lot of the structured data. You can also use rsync and FTP. So when downloading data, when downloading structured data, it's important to be mindful in selecting what data format to use. Of particular note, it's important for us to emphasize that the current standard file format for data in the PDB archive is PDBX mmsif, which is data with the extension .sif or .sif.gz. And this is the official format, and users should use this. And if they're not using this already, begin to migrate to using this. For those who have used or worked with data from the PDB in the past, chances are you're familiar with the legacy PDB format file or with the .pdb extension. And I suspect many people may still be using this, but since 2014, we've stopped supporting extensions to this format in favor of the PDBX mmsif format. So in this new format, data is saved as a set of category and attribute pairs along with their corresponding values. And these categories and attributes are all defined in an authoritative dictionary, which you can find by browsing this site here. So this new format offers several benefits over the legacy PDB format. The first is that it supports structures that have more than 100,000 atoms, which is a limitation that the fixed column width restrictions of the PDB file format can't handle. The second major benefit is that it's extensible. And when I say that, I mean that when new experimental or computational methods arise that require their own type of metadata to define that type of experiment or to define that type of software, we need to add new categories and attributes in order to describe those new techniques. And so through this new format, we're able to do that because we can simply add new definitions to the dictionary to define those new types of metadata. So this is one of the main motivations for why we have transitioned here. So now as we begin to enter the tutorial part of the talk, let me begin by first introducing the case study that we'll be focusing on, and that is of the Arabidopsis thaliana resistosome. So Arabidopsis thaliana is the gold standard model organism in plant molecular biology. I'm sure that all of you at some point in your studies or career have come across Arabidopsis in your textbooks or your research. And the reason for that is because it's such a well-studied and extensively characterized organism. And like all organisms, it also maintains an immunity or immune system, where it has to fend itself off of pathogens from the environment and maintain its survival. And one of the main ways that it does this is through the formation of the resistosome, which is shown here. So the way that this forms is through the sensing of a pathogen by a structure known as a nucleotide binding leucine-rich repeat receptor. And upon the sensing of these pathogens, these receptors oligomerize into a pentamer, which forms the fully active resistosome as illustrated. Ultimately, this process culminates in cell death. And the working hypothesis for how that happens is that this resistosome may embed itself into the plasma membrane of the cell and then create a pore and thus disrupting the plasma membrane. So the steps that it follows in forming the resistosome are illustrated in this schematic here. First, the NLR begins in its inactive state in which it's bound to ADP. Upon indirect pathogen sensing, it then undergoes a conformational change where it releases ADP, and then it undergoes another conformational change where it binds to ATP. Once it's bound to ATP, it then oligomerizes into the pentamer fully formed resistosome. And so for purposes of the tutorial, I thought it would be helpful if we tried to actually find some of these structures, or at least the corresponding structures for these steps in the pathway, which have been published by Wang et al. And so when we do this, we can keep in mind some of these key elements of this pathway in developing our search. So for example, we can use the fact that this first inactive state is bound to ADP to help narrow down our search. We can also use the author and citation information. We can also use the fact that it's coming from Arabidopsis thaliana. We can also use the fact that these structures were determined by electron microscopy, which I found out by reading this paper here, and as you would be able to as well. So let's go ahead and try to do this. So I'm going to exit out of my presentation mode, and I'm going to go to rcsb.org. And so when we land on rcsb.org, we are presented with the top bar search. And so let's go ahead and just try to use that. So let's try to just search for Arabidopsis thaliana. And you'll notice that as I type, there's some suggestions that start populating in the drop down below. And if you were to click on any of these, it would allow you to perform a very specific attribute search for this specific keyword. But for purposes of the demo, I'd like to just perform a general full text search, and you can do that by hitting either enter, or you can press the search icon. And when you do that, you get approximately 2,400 structures. You can change the number of structures that are shown on each page by selecting from this drop down here. You can also change how these are sorted by default, they're sorted by relevancy score, but you can sort them by date, as well as other criteria. On the left, you have the refinements panel where you can see what type of structures are included in this result set. So these are some popular properties of structures that people like to refine upon. So they are included in the refinements panel. So although we search for Arabidopsis thaliana, we see some results from Homo sapiens, as well as other organisms. And that apparently must be because we performed a full text search in which these structures at some point in their metadata also include the words Arabidopsis or thaliana. So obviously we want to filter this down, and we want specifically Arabidopsis thaliana. So if we go ahead and check that and then click the refinements button, we've reduced our structure result set to about 1,800 results. And when we scroll up into the advanced search query builder, we can see that this property was added to the advanced search builder. So if you're not sure on what property to select, you can always select it from the refinements panel. But obviously we have to filter this down a little bit more. So let's try to add another attribute, and let's try to filter this down on the experimental technique that was used. So all we have to do is click add attribute. When I click that and I click this, you can see all the options that you have for structure attribute search. You can search by entry ID, deposit ID, structure title, author, release date, as well as methods, experimental method. And if you're not sure, and obviously this is quite extensive and it might be difficult for you to manually go through every single one of them, you can always just start typing it in. So you can just type experimental and you'll see that experimental method would show up. So you can click that, and we know that the method was electron microscopy. And if we hit search, we've reduced our, oh, I'm sorry. I wanted this to be an AND operation. Hit search. There we go, voila. So now we reduce our result set all the way to 100 structures. So obviously though, 107 is still kind of a lot to go through manually. So let's try to add some more filters. The next one we can add is the author. So the author of the paper is Wang J. So let's add another attribute. And let's look for author. It's right there. And we can type in Wang J. So we hit search. Now we're down to 10 structures. It's great. So still 10, we can narrow this down even more because we're trying to find that one specific structure that has ADP bound. So how can we do that? We can use the chemical attribute search. So let's open up that part of the advanced search builder. And we can select chemical IDs under chemical components. And you see it even provides some example queries of what you can use for values. This is to help users try to figure out exactly how to format or what type of values that the property is looking for. So we can type in ADP. And if we do this, we're down to 2 structures. And so now from these 2 structures alone, we can quickly tell which one's the right one. And the way we do that is we can simply use the citation information. So this is the 2019 paper from Wang J et al. So we have successfully found our structure. And when we click on that, we're brought to the structure summary page, which contains some general information about the structure, as the name implies. It provides the title of the structure, some information about where it came from, when it was deposited, experimental data, validation information. You can also download the structure directly from the page. So if you were to click on this dropdown, you can download the FASTA sequence, the PDBX MMSIF data file or the compressed file, or the PDB format file. But again, please try to begin migrating to the MMSIF as this will continue to be supported moving forward. You can download other related information. We also link out to the corresponding experimental data at EMDB. And if you scroll down further, we have the citation information, and then we're provided with a breakdown of the structural contents of the entry. So as every entry contains at least one or more protein chains, this is a breakdown of each of those unique chains. So an entity is a unique protein chain. And so the first entity is RKS1, and the second entity is disease resistance RPP13 like protein 4. So this contains 2 unique chains. And if we scroll down further, we see what small molecules are included here. And as we search for it, it contains ADP. And then we have more information about the experimental and validation, as well as the deposition history and revision history. If we scroll back up, we'll see that there's a number of different tabs up here on the structure summary page. Each one brings you to a different aspect of the structure. So 3D view would bring you to a 3D view of the structure where you can actually interact with it, play around with it, click on it, and this will be demoed more thoroughly by Shuchi later on. So I'm going to go ahead and go to annotations, and this illustrates some of the externally integrated annotations that we provide for our structures. So this provides annotations at an entity or protein chain level. And as you see, we have some ECOD annotations, protein family annotations, gene product annotations. And then we can also go to experiments, where we're provided with information about the experimental conditions used to solve the structure. to determine the structure. And if you go to sequence, you get a overview of residue-specific annotations. So you can see the level of disorder for different residues. And sorry, I kind of did that without explaining. You can zoom in by scrolling. So if you zoom in, you can see the specific residues. And if you hover over any of these in the upper right, you see the information about that annotation. And then you can even select what chain you want to look at. So if I selected chain B, we see we have most of the same annotations, but in addition, we now have this additional row for binding site of ADP. So this signals to us that the entity that we were looking for is entity 2, because this is the one that binds to ADP. This is the NLR. When you, just to complete this story, when you go to the genome tab, we have genomic information about the entry. And we also provide version information about the entry. So like when any changes are made. So now that we know that the corresponding entry or entity is the second entity of this structure, that is the one that's bound to ADP, we can scroll down here and we can see that we're provided some options to link out to UniProt searches. So by clicking on this link here, you could search for all structures in the protein data bank that have this UniProt ID. And this will be what we want to do because we know that in this pathway, that NLR never actually changes its sequence. The sequence remains the same. The only thing that changes is its structure and what it's bound to. So we can use that as a way to find the remaining structures in that pathway. So if we click on this, and I'm going to open up a new tab, we see that we're presented with 5 structures. And scrolling through them, we see that they're all from the same literature citation and from the same authors. So this represents the complete set of structures that we originally set out to look for. And now that we have these, if you're interested in downloading them, you can choose to either click on each one individually and download their structure files from there. You can download the structure files by clicking on this button. But even more efficiently, you can click on this button here, which will download all selected files. And if you click on that, it'll bring you to our batch download tool, which lets you specify multiple structures to download the structure of. And here you would just select PDBX MMSIF as your data format, and then you would generate the file for batch download. I'm going to go back because I'd like to also describe some of the other links that exist on the structure summary page that I kind of skipped over. You'll see these plant protein or Arabidopsis thaliana. Any of the links that you see on the page will, most of them will let you perform a search for that particular structure attribute. So if you're on this page and you wanted to then look at all the structures for Arabidopsis thaliana, you could click on this as the organism. So that's another helpful feature about our summary pages is the ability to link out and perform different types of similarity searches. You can search for structures by similar 3D structure or sequence. So we offer a variety of options for facilitating searches throughout our site. So I wanted to go back though to this search page that I had. All right, so we kind of developed this rather complex query. How would we do this through programmatically? Well, the simplest way to do that would be to click on this JSON button as I described. If I click on this, it brings us to the RCSB PDB Search API Query Editor. And then it automatically populates that editor with that corresponding JSON for the query. And if you press this play button here, you get the results that we just had on that page. You get a list of the identifiers along with the corresponding relevancy score for those structures. By default, these searches are restricted to the first 25 results. So if you want to get more results, you can either increase the row number or you can continue to paginate, which is the more programmatically friendly way of querying an API. So along with this programmatic search query option, I also wanted to just quickly illustrate how you can access our PDB archive directly to download files. So you can go to our documentation page and go to programmatic access. And here we provide a lot of documentation about how to download our data. And one of the things I described earlier was that we now offer our data on an AWS S3 bucket, which you can access here. So if we open this up, this will bring us to the PDB archive. And if you're looking for a specific structure, and let's say we're looking for the one that was on our search results page, let's say we're looking for 6J5W, we would go to under public, PDB, data, structures, divided, MMSIF. And obviously you can see we offer all the formats on here, and we offer a lot of different data like NMR data and so forth. But for purposes of the tutorial, I'm just kind of quickly navigating through to find the specific structure. And again, this is being recorded, so I hope that you'll be able to review it later in case you miss anything. So we're looking for 6J5W. So to get there, we need to use the 2-letter hash, and that's going to be the middle 2 characters. So it's going to be J5. And so we can even just search for it here. If we type J5, and then we were looking for 6J5W, 6J5W. And voila, we have our structure. So we can click on that. It'll download the structure. And now you've been able to use our PDB archive directly. And again, you can use your own curl or Python scripts to perform these downloads as well, which is probably the more useful way of using our PDB archive directly as opposed to navigating manually. But you can always navigate it manually if you wish. With that, I just wanted to provide a quick summary of what we did. I illustrated some of the interactive methods that you can use for searching data and downloading data, as well as some programmatic methods you can use for searching our data and downloading our data. And then I wanted to provide some important links that you might find helpful when you want to try to follow along with any of the things I demonstrated. And with that, I'd like to thank you for your attention.